Hi, everybody. This is Dr. Lonneville. This is online podcast number 8, the last one of the semester. Hooray. And then I'll talk to you separately about the final. Okay, so in online podcast number 7, we started to talk about cancer, which has a lot of the properties of incorporating stuff you've learned about pathways and techniques up until this point in the course. This podcast, the second one on cancer, is going to talk about specific pathways that we've talked about and where things are going wrong in cancer, and hopefully give you some more context about why we were talking about those pathways. Normally, I would have a third lecture on cancer where I talk more about some of the macro statistics. I'm going to put a little bit of that in this podcast, but this semester, certainly a weird semester, spent more time on COVID-19 and on the molecular immunology than I normally would. And here we are. Okay. So if we look at the type of proteins that are typical for oncogenes, again, oncogenes are genes that code for proteins that you need to have in their wild-type form, but those genes tend to accumulate mutations and break, and when they do accumulate those mutations and break, they code for something that causes cancer. And there are many oncogenes out there, and they come in different classes. So you could have oncogenes that produce the signaling molecule, the hormones that we've talked about, the first signal. We have oncogenes that code for the signaling receptor. We can have oncogenes coding for the intracellular transducer, what happens after the receptor dimerizes, for example, or the receptor binds its ligand. You can have oncogenes code for intracellular receptors. We haven't talked a whole lot about intracellular receptors, but there are some intracellular receptors in pathways for hormones that are lipid soluble, like testosterone, for example. It binds to an intracellular receptor. There are oncogenes that are transcription factors. We've talked about a lot of transcription factors. You can imagine how if that transcription factor binding is somehow altered by a mutation and you turn on genes when you're not supposed to, or you don't turn it on when you are supposed to, you can imagine how that could lead to uncontrolled cell growth. There are oncogenes that are involved in DNA repair, so that if you can't repair mutations which are just going to accumulate from any organism interacting with its environment, and if you can't fix those, then they can lead to uncontrolled cell growth. And then cell cycle control proteins, we talked about cyclins and cyclin-dependent kinases participating in cells making decisions about, should I go into the S phase, should I leave G1, those proteins are mutated, cell is no longer making those decisions correctly. And then the default has been that, if things go really wrong, we'll just kill the cell by apoptosis. But if the proteins to make that decision are mutated, they can lead to uncontrolled cell growth also. So that there are lots of these and lots of these and lots of these and lots of those and so on, because we've talked about just a smidgen of them, and there are lots of them out there. So if all those things can cause cancer, there are certainly lots of oncogenes out there. So one thing about oncogenes that seems to be true, has been shown in lots of studies, is that they cooperate in terms of the uncontrolled cell growth phenotype. So if we look at an oncogene like MYC, just the name of an oncogene. And we look at how likely is it to cause a tumor if this oncogene is expressed in mice. So mice usually live out to, say, 200 days. And if they are expressing MYC, their life is cut short by 80% of the mice, or 20% of the mice with a MYC mutation have a tumor by, say, 180 days. And if we look at RAS by itself, this is RAS as an oncogene, they start getting tumors earlier, and more of the mice have tumors by, say, day 180. But if we look at MYC and RAS together, it's not just the incidence of MYC plus the incidence of RAS, it's worse than that. And that if MYC and RAS are expressed together, you get a multi-hit effect, and that it's much worse for the mouse than one oncogene being expressed. And unfortunately, one oncogene being expressed can make it more likely that a second oncogene would be expressed. Remember, in these classes of molecules that get messed up, if you mess up DNA repair protein, for example, that could literally fix mutations, and now that DNA repair protein is knocked out, then your ability to fix mutations can be eliminated, and so therefore the chance of mutations persisting goes up. Some of those mutations are oncogenes. So oncogenes beget oncogenes. Okay, some specific cases that we have hinted at in the past. I told you cause cancer. HER2. So HER2 is a RTK receptor. Remember, there are different isoforms of HER1, 2, 3, 4. And what happens in HER2 oncogenes is they form these heterodimers. So it's usually supposed to be HER2 with another copy of HER2 to form a dimer. But in heterodimers, you can have HER2 dimerizing with a HER1 or HER3, whatever. And what happens is these receptors tend to stay. This is not HER2, this is HER2, and this is JAK-STAT because it's EPO. But what happens is in oncogenes, the signal tends to stay on either constantly or more often than it should, longer than it should. And since these tend to be growth and stimulatory in nature, you get more of that than you should in nature. Because this is a well-known mutation in female breast cancers, there have been targeted biologics, monoclonal antibodies, developed as treatment against breast cancers, and they've been really successful in treating some breast cancers. So if you know someone who's ever had breast cancer, the breast cancer is typed in whether or not it's HER2 positive or HER2 negative, estrogen sensitive or not. This is a characterization of the tumor itself and how it responds, what proteins it's expressing, essentially. And those proteins that are expressing HER2, mutant HER2, are called HER2 positives, and those tumors can be treated successfully, usually with antibodies against HER2. So those antibodies are injected into the patient. They attach to cells that are expressing HER2. Remember, it's a membrane-bound protein, so this extracellular domain would have an epitope that would recognize that antibody. That antibody tags that cell, and then parts of the innate immune system, natural killer cells, macrophages, will recognize that antibody and kill that cell. This is much better in terms of side effects for the patient because more old-fashioned or traditional chemotherapies just go after cells that are rapidly dividing. So cells that are rapidly dividing could be cancer cells, or they could be cells that rapidly divide as part of homeostasis. So hair follicles rapidly divide. The lining of your gut from your mouth to your anus rapidly divides because you abrade away those cells daily, and those cells are replaced by mitosis. So if you have a general chemotherapy agent that kills cells that are rapidly dividing, something like tamoxifen, then you can have really debilitating side effects like nausea and hair loss. Whereas these more advanced or more specific chemotherapies, like the HER2 antibodies, only attack those cells that are expressing HER2 mutants and ignore the cells that are still wild type, still doing their job, and don't kill those cells. And so much lower side effects. Okay, here's an H5P question for you. All right, RAS, we've talked about RAS a lot in this class. RAS is a common oncogene. If you just switch out a valine for a glycine in RAS, it becomes RAS-D, which is an oncogene. And that this oncogene has reduced GTP-ACE activity, okay? This molecule right here. And therefore, the on form, where GTP is bound, stays on longer than it should, because the GAP, which would hydrolyze that GTP, convert it into the off form, inactive RAS, has lower activity. And so RAS stays on longer than it should. We talked about RAS in this pathway when we talked about our first kinase cascade. RAS, through adapter proteins, which they're not showing on here, activates RAF. Remember, RAF is normally bound to 1433. RAF activates MEK. MEK activates a MAP kinase. And then phosphorylates a transcription factor that's going to bind to DNA and change what genes get just transcribed. In this diagram, all the proteins that are in dark green here are proto-oncogenes in that they can become oncogenes if mutated. So the receptor, all the adapter proteins, RAS itself, everything in the kinase cascade, all possible oncogenes. And then you have a protein which we haven't talked about before called NF1, involved in RAS interacting with RAF. It can actually act as a tumor suppressor gene. But see, in this pathway, it's sort of daunting in that anything in this pathway is a possible oncogene. And this is typical in these signaling pathways. Signaling pathways that are involved in cell growth, or cells making a decision whether or not they should stay alive or go through apoptosis. These signaling pathways, as you well know, involve lots of proteins. And anything in that pathway could be an oncogene. Another sort of thing that we haven't really talked about that can cause production of an oncogene is translocation. So, in other classes, in cell and molecular class, and in principles, and in genetics, you talked about how there's crossing over during meiosis, where homologous chromosomes will switch parts of their chromosome arm from one chromosome to another. In this is not during meiosis, but just during cells being cells. You can have unrelated chromosomes switching off parts of their chromosomes' arms. And So these two chromosomes, chromosome number 9 and chromosome number 22, are switching off parts of their chromosome. So part of 22 becomes part of 9, and part of 9 becomes part of 22. And where they switch is right at a reading frame for a protein, and that results in a protein that is what's called a fusion protein, part this, part that, and it translates into a kinase, and this kinase is constitutively on. So all the kinases that we've talked about in wild type, they can be switched on or off, sometimes by another kinase and off by a phosphatase. This kinase is always on. And if it's constantly phosphorylating things versus other kinases that can be on or off for a short amount of time, you can imagine how that could be bad. There's been a drug developed called imatinib, which is a competitive binding site for a competitor for the binding site on this kinase, this mutant kinase that gets made. And so it would have normal substrates, quote unquote normal substrates that it's going to bind to that can get phosphorylated, say like a JAK or a STAT that it could bind to and phosphorylate, and it has a certain shape. Imatinib is a drug that has a similar shape to the binding site, and so you flood the cell with this drug and it outcompetes this binding site so that the normal binding sites are not phosphorylated. Sorry about that. A little brain stumble there. So it's been a super successful drug. I know some patients who have been on it for years with very few side effects, and really has taken their cancer to task. Okay, here is another chromosomal translocation. This one that I talked about here is between chromosome 9 and 22. Here's a different one, chromosome 8 and 14. Switch off parts of their chromosomes. Results in a cancer called Burkitt's lymphoma. MYC, which we talked about in the first slide as an oncogene. Normally it's a transcription factor, normally expressed at very low levels, and transiently expressed, you make a little bit of it and it goes away. In Burkitt's lymphoma, this chromosomal translocation, the gene that gets transferred in front of a very active promoter, one that normally makes antibodies, so it's expressed at a high level and it's expressed often. Now you have this transcription factor that's made like antibodies are made, which is not a good situation, and so results in lymphoma. Okay. Some other things that we have talked about. So we talked about RB as an inhibitor of a transcription factor called E2F, and that RB gets phosphorylated in order to stop inhibiting E2F, and then E2F goes ahead and binds to DNA to turn on genes that are required for the S phase, like DNA polymerase, for example. The proteins that are involved in phosphorylating RB, as well as RB itself, can all be oncogenes. So you have P16, which is inactivating this cyclin-dependent kinase 4. This cyclin-dependent kinase 4 depends on the cyclin D, P16 is shutting off this kinase. So if P16 is mutated, this kinase can now be active, and RB can be phosphorylated more than it should, and therefore it's not inhibiting E2F as much as it should, and it sends E2F to the nucleus more often than it should, giving the signal to enter the S phase more often than it should. So again, it's not just RB that can be mutated, and I mentioned when we talked about it in the last podcast, it's mutated in almost all cancers. But anything in the pathway that interacts with RB can also be mutated and lead to an oncogene, or lead to cancer. We talked about TGF-beta for the last test as one of the pathways that control cell growth. In the case of TGF-beta, it's an anti-growth pathway. And so if you have a mutation in the pathway, then the pathway is not working normally, and therefore the cell is getting the signal to grow instead of not grow when it normally works. So this is another example of, this is a very complicated pathway with many steps in it, and any of those steps can lead to cancers. So you can have mutations in TGF-beta itself. You can have mutations in the SMADs that interact with the receptors when they come together. You can have the mutations in proteins that are involved in getting the SMAD into the nucleus. You can have mutations involved in the DNA where SMADs bind to the nucleus. So once again, it's one of these pathways. It's involved in cell growth. Anything in the pathway that is mutated could result in uncontrolled cell growth or cancer. Here's another H5P question for you. Okay. Any sort of superficial dive into the molecular biology of cancer, we'll talk about P53. And we're talking about molecular biology of cancer, hopefully not in a superficial way, but what I'm saying is, if you do any kind of literature search on how does cell biology or how does molecular biology, what do we understand about the molecular biology of cancer, you'll come up with the protein P53. And P53 is another one of those super common oncogenes. And how it is involved in cancer is, in one sense, pretty simple, and in another sense, more complex. Okay, so normally what happens is P53 is made and then degraded. Okay, made and then degraded, made and then degraded. So P53 concentrations are typically low in a normal wild type cell. Okay, made and then degraded, made then degraded, degraded by ubiquitin. Okay, and ubiquitin tagging. When there is a problem, okay, a problem during G1, P53 is then stabilized so that it's no longer ubiquitinated. So if P53 is phosphorylated, it is stabilized and no longer a target for ubiquitin. And stabilized P53 is a transcription factor that will bind to DNA. And when it binds to DNA, it can synthesize DNA repair mechanisms. So it'd say, okay, there's a problem. There's mutations. Let's try to fix those mutations. Okay. At the same time, it'll stop the cell in G1 or G2. Okay. But most of the time, we're talking about the transition from G1 to S. So P53 is saying stay in G1, give yourself a chance to repair. And if it can't be repaired, if the cell can't be repaired, activate apoptosis. Okay, so we talked about PUMA when we talked about apoptosis, and PUMA is released to stop inhibiting the oligomers from forming a channel to release cytochrome C, and cytochrome C is released, starting the cascade of caspases that will lead to apoptosis. Okay, so normally p53 is made in a normal cell and kept at low concentration by ubiquitinylation, okay, by interaction with MDM2. If you want p53 to actually be a transcription factor, and there are enough of it to be around to be a transcription factor, you have to stabilize it. You stabilize it by phosphorylating it, and phosphorylated p53 can then accumulate and go into the nucleus and stop cells in G1 before they go into S, and also repair the DNA, transcribe genes that are going to repair the DNA, and if not repair the DNA, activate PUMA, which is going to activate apoptosis and kill that cell. Okay? So again, normal healthy cell, you don't need p53. Okay? Nothing's wrong, you don't need p53. And mice that have p53 knockouts can reach adulthood, but they tend to accumulate tumors because they can't activate the mechanisms to repair their DNA. So if nothing goes wrong, you're fine. If something does go wrong, you're not fine. Okay. How do you know if something has gone wrong? Okay. There are monitoring systems like ATM, okay, which is a kinase. And ATM specifically detects double-stranded breaks in DNA, and ATM can activate mechanisms to fix the problem or kill the cell. Okay, and how do they do that? 3 different mechanisms. ATM can change the phosphorylation of CHK2. CHK2 can change the phosphorylation of CDC25A, and that will get degraded and lead to a very rapid G1 block, S block. So you don't leave G1, or you stay in G1, and you're prevented from entering S. Okay, so these are proteins that would be needed to progress out of G1 and S. They get degraded rapidly, and so you stay in G1 and you're not allowed to enter S. So that's one mechanism by which ATM can keep things in S. The other mechanism is ATM can directly phosphorylate p53 and stabilize it, so p53 builds up. The third mechanism is it can phosphorylate MDM2, and MDM2 can now no longer tag p53 and cause p53 to be degraded, so p53 builds up. Okay, this is a mouse-specific pathway. Don't know if this occurs in other mammals, so we're not going to talk about that. Okay, so ATM, 3 different pathways can keep cells in G1, prevent them from entering the S phase. 2 of those 3 involve stabilizing p53. Okay, here's a nice little quote. It seems nearly impossible for a normal cell to become a cancer cell unless it activates the p53 network. Okay, and by that meaning there's some problem. So you can activate the p53 network and fix your DNA, and then you're not going to be a cancer cell. But something went wrong in p53, which is a cell repair mechanism or a cell fate mechanism, in that it's going to kill those cells if it's non-repairable. Okay, so if you can't do this function, you enter S when you should not, or that cell survives when it should not and could lead to cancer. So ATM detects double-stranded breaks, and it monitors for those double-stranded breaks, and then it can affect the activity of CHK1 in one pathway, stabilization of P53 in another pathway. All right. More about P53. So P53 is this transcription factor. It's going to bind to DNA. Here's DNA right here. Here's P53 binding to the groove in DNA. It is a tetramer. There are 4 subunits to P53. If you have multiple alleles for P53 and only one get mutated, that one mutated allele is still making a P53 monomer. That monomer is going to try to form a tetramer with wild type P53, ones that aren't mutated. And if only one out of the 4 is mutated, it still results in a mutated tetramer of P53. So that you can have like one bad apple among your P53 genes and will still mess up a large proportion of your P53 protein. Okay? And then even more complexity about P53. So if you get into this and read about P53, it can get really confusing because you're thinking that P53 is a good thing, but sometimes some papers are saying it's a bad thing. So what's the deal with that? Okay. So if P53 becomes an oncogene and acquires a mutation, it not only just loses its ability to turn on genes that will stop the cell in the S phase, stop the cell before it enters the S phase, stop the cell in G1, also turn on genes that will repair the DNA. So it not only loses that, but it gains function as an oncogene where it turns on bad stuff. So it's not only not turning on good stuff, but it's also turning on bad stuff. Okay? So if we look at wild type P53, so no mutation, okay, it has a role in organogenesis, in differentiation of cells. Okay? In embryonic stem cells, you have very low P53. But as stem cells start to become differentiated, start to acquire their cell fate, okay, they have higher concentrations of P53, which is required for those cells to differentiate, okay? This is what's called a cell fate landscape in 3D. And if you think about this as marbles rolling down a hill, and you start with a marble here, it could go this way, it could go that way. If it goes this way, there's these 2 little pathways that it could go down, and they are the most likely to go down just because of the shape of this landscape. Okay, it's not likely that it's going to roll off this way. Hopefully you can still see my arrow on the podcast. What I mean is to the right as you're looking at it off the slide. These are the most likely cell fates. Okay, and so there's a balance between self-renewal and differentiation. Once the cells become differentiated, then P53 becomes really low concentration again. As we said in the last slide, it's normally low concentration. But while cells are differentiated, you need a relatively high concentration of P53, and that's part of its normal function. In mutant P53, over on this side of the landscape, okay, there are lots of amino acid sites, lots of codons on the P53 gene that are mutated. But 6 in particular are so-called hotspots. And they will gain function as they are more likely to form tumors than if you just got rid of P53. So P53 has multiple codons. Multiple codons acquire mutations and can lead to a mutant P53. Some of those, 6 of those codons, not only get rid of the normal function of P53, but also add a function in that not only am I going to not do what P53 normally does, but I'm going to make tumors at a higher rate than I would even if I didn't have P53. So they turn into tumor makers if you get those 6 hotspot mutations. So the previous understanding of P53 is that you normally don't need it as an adult cell once you're differentiated, and only if something goes wrong do you need it. And therefore, if you have a mutation in it, you can't activate DNA repair mechanisms, you can't activate stopping the cell in G1, and that can lead to cancer. This new understanding of P53 is that for cells as they're developing, as they're turning in from a stem cell into a liver cell or a stem cell into a bone cell, for that differentiation to happen, they need a relatively high concentration of P53, and that's part of normal P53 function. Also, that among the mutations that P53 can accumulate at 6 codons, if those amino acids change at those spots, it turns into not just I don't have P53 function anymore, but now I have a gene that will actively generate tumors. So not passively by not stopping cells from accumulating mutations or killing cells that have accumulated mutations, but actually causing tumors to grow at these 6 spots. So that's why if you read some newer papers on P53, you're going to see some information that seems confusing in terms of, well, I thought P53 was good, and they're saying it's bad. It depends on if they're talking about a mutant P53 that has one of these 6 hotspot mutations. Okay, other factors can indirectly activate oncogenes. Certainly environment and exposure and life experience can have a great impact on cancer formation. We know that from identical twin studies. Some of those twins are recruited at the Twinsburg Twin Festival that has a high number of twins showing up to that, and so they'll recruit it for these clinical trials. And from those sorts of studies, we know that only 20% of identical twins share breast cancers and types of breast cancers. So those are 2 individuals that are clones of each other. They have the exact same DNA, but they haven't had the same environmental exposure over the course of their life, and therefore a large percent of what determines whether or not someone's going to get cancers depends on what kind of environment they've experienced during their life. In that environment can include viruses. We've certainly talked a lot about viruses this semester. If for DNA-based viruses, the viral DNA is inserted specifically or randomly into the genome, and it may be incorporated at a spot that causes higher production of a protein or lower production of a protein, or activating a kinase or inactivating a kinase just by where it gets inserted. RNA viruses, including SARS-CoV-2, which is a RNA virus, they are, or sometimes, but not always, integrated into the DNA genome by being reverse transcribed and then incorporated into DNA. I haven't seen anything about SARS-CoV-2 where that is true. SARS-CoV-2 is a plus RNA virus in that its viral genome is just like mRNA, and then the mRNA is translated by our ribosomes to make more copies of itself. But anyway, for RNA viruses that do incorporate into the genome, those are called provirus when they're incorporated into the genome. So converted into DNA and then converted into the genome. And then once it's incorporated, the genome can be passed on to progeny. Again, I'm not saying that about COVID-19. Okay, I'm talking about other viruses. There's different estimates of how much of a total genome is once originally viruses. In mice, the estimate is as much as 1% of the total genome is proviral. It's thought that the transcription factors and remnants of transcription factors, highly repetitive DNA in our own human DNA, was once viral, had a viral origin. And maybe we're just carrying that DNA around and it's not doing any damage to us. But there is evidence that viruses, once incorporated into a genome, or just by the going through their life cycle, not necessarily incorporated into a genome, but just by going through their life cycle, can cause cancer. So these include HIV, hepatitis B, human papillomavirus, HPV, Epstein-Barr viruses. These all can cause cancers in humans. Some really well-known cancers. So, for example, HIV positive people are susceptible to a cancer called Kaposi sarcoma. Okay. How much of cancer is due to the environment? This is kind of an old review. I apologize, 2008. But it just has this easy to understand graphic, which is a pie graph, saying that 90 to 95% of cancers are estimated to be initially triggered by environmental exposure, whereas only 5 to 10% of cancers are exclusively genetic. So there's always going to be an interaction between environment and genes. So you can't say that even in these environmental cancers that there's no involvement of genes. What they're talking about is the 5 to 10% is that you just inherited from your biological parents genes that gave you increased susceptibility to cancer, and they're talking that's about 5 to 10% of cancers. Environmental cancers, those that have a strong environmental component, and again, you can't say it's all this or it's all that, but in trying to identify factors in the environment that increase the likelihood that someone will develop cancer, those include diet, tobacco use, infections like we just talked about, obesity, alcohol use, and other factors. One very well-known association is with cancer and smoking. Male smokers are 23 times more likely to develop lung cancer than non-smokers. Female smokers are 17 times more likely to develop lung cancer than non-smokers. This may have to do with the difference in immune systems between males and females. It may have to do with socioeconomic factors and just society factors, like females are typically more likely to go see a doctor regularly than males are, but males are more susceptible than females, and certainly both much more likely to develop lung cancer if they smoke than if not smoking. I want to go to the cancer.gov site here and show you some stats. Just to be some up to date on some of these things. So this is cancer.gov, the Cancer Institute, as part of the U.S. government research agencies. 1.7 million new cases in the U.S. In 2018. 600,000 people in 2018 died from cancer. In the U.S., the most common cancers are breast cancer, lung cancer on the top, but that'll differ by country. I asked you an H5P question about that, and that's because different factors that affect cancer, like genetics, like environmental exposure, like the age profile of the population, those are different in different situations. In the U.S., breast cancer is number one. Okay, just a couple things here. Kids are getting cancer. There are 15,000 cases out of those 1.7 million, so relatively rare. It costs a lot of money to treat cancer in the U.S. Worldwide, it's among the leading causes of death. If you are in a less developed part of the world, you're more likely to develop cancer and to have a worse outcome than if you are in a more developed part of the world. But overall cancer death rates in the U.S. have fallen over the past 30 years by 26%, and that's pretty remarkable. So when I was a kid, which is close to 56 years ago now, if you heard about somebody getting cancer, that was a death sentence, and that is definitely not true anymore. So our ability to detect cancer and to treat cancer has improved dramatically. We are not at the point where we say we have a cure for cancer, but there are cancers that can be cured, and that the person has no detectable cancer after being treated. And certainly what we know about how cancer works has tremendously affected our ability to help people survive cancer. Okay. If we look at Ohio and how we compare to the United States, we have a higher rate than the U.S. on average in Ohio than in the United States, depending on the cancer type. Most of these we're higher on. Some of these are related to diet, like colon and rectum cancer. We have a high rate of obesity in the United States, and so we are at a higher rate of colon and rectum cancer. We are smokers in Ohio, and so have a significantly higher rate of lung and bronchus cancer compared to the rest of the United States. And overall, our rate is higher in Ohio than the rest of the United States on average. Okay, environmental exposure if we use epidemiological tools like they're trying to do right now with COVID-19 exposure to just look like how many people are sick and when are they getting sick, we can get some ideas about what is happening. So carcinogenesis is development of cancer. Chemical carcinogenesis is chemicals that cause cancer. If we look at the per capita cigarette consumption, it peaked in the mid-60s in the U.S., and about 30 years later, you get a peak in male deaths from lung cancer, and because that's about the time lag here, you can see that the female lung cancer death rate is following that. It's going to peak about probably in the 2010s, and it's going down in the U.S. The cigarette companies, when they pass legislation that you can't advertise in the U.S., can't advertise on TV, for example, they started going after other markets in other countries like China and Eastern Europe, and they are now seeing these sorts of curves in those countries. So don't smoke. I mean, it's a very well-established cancer risk and very obvious cancer risk. Smoking marijuana is also a risk, but it's a qualitatively different risk. But you're still at risk for lung cancer. Bob Marley died of lung cancer and was a big marijuana proponent. Other risks. So this is Henrietta Lacks. She died of cervical cancer at Johns Hopkins Hospital. Dr. John Gay cultured her tumor. He attained the first immortal cell lines. So when we talked about cell strains versus cell lines, he named them HeLa cells from the first initials of her first name and the first initials of her last name. These cells are super robust, really hard to kill. They can be mailed in regular mail. They don't have to be refrigerated. They have taken over other cell cultures over the world. So labs that work with HeLa cells and also work with other cells found out that the HeLa cells were taking over all their cell cultures. They can spread through the air. These were this cancer derived from the human papillomavirus, which Henrietta Lacks likely got from her husband, and she had this large cervical cancer tumor, which was the generation of these cells. Her identity was kept secret until 1975, but then lots of patents and technologies have resulted from using these cells in the lab. Her family is very poor, and they have never received royalties or any kind of official recognition from the contribution of her cells. Even recently, the scientists have been very tone deaf about how they've treated this, and not thinking that there's an actual person or people behind these cells. They published a genome to the scientific community just a couple of years ago of HeLa cells, which are very unusual cells, in that they're cancer cells, they're polyploid, they have all kinds of weird stuff going on, and they published a genome of those cells. And didn't consider that there are actual relatives of Henrietta Lacks out there whose genome would be very similar, and I wouldn't want my genome published and attached to me, and they didn't want to either. And so the NIH retracted that genome information after it had been out there for a while. And Francis Collins, who's the head of NIH, went to apologize to the family. But there's been all kinds of scientific deafness about the human side of this scientific story. Some of the good that has come out of this is that Merck developed a vaccine against the human papillomavirus, the virus that causes this cervical cancer. They have their vaccine is against the four most common strains that cause HPV and cervical cancers and genital warts. They, but not all strains of the virus. It has been really effective in reducing the incidence of cervical cancers and genital warts. There are some countries that are saying they are close to wiping out this disease, which is tremendously great advance in science. It started off in girls, then in 2009 was approved for boys. There have been some very serious side effects from the doses of this virus in girls. Some 6,000 girls have had serious side effects, neurological effects, effects at the site of injection. It's a very low incidence among the doses that have been given, 16 million, but it's not a small number of people, 6,000 people. So this is not an anti-vax thing at all, but for people who are considering getting this vaccine, they should look at risk factors, if any have been discovered for people getting this vaccine, and consider that if they're a good candidate for this vaccine. Certainly a very effective vaccine, but like with all vaccines, has risk, and that includes this one. Okay, how do carcinogens, chemicals that cause cancer, how do they cause cancer? So they can directly mutagenize the DNA. So they can attack your DNA and change the chemical composition of your DNA, and therefore make it difficult or impossible for that DNA to be replicated. Or they are metabolized into something that can become mutagenic. So here are just some pathways about how these toxins can cause mutations. Aflatoxin is a mold on grains. So grains that are stored in silos, which are most grains in this country, can accumulate aflatoxin, and that can be a very powerful mutagen. Just as an example of a chemical carcinogens, this is a quote from a environmental toxicology paper talking about chemical carcinogens, just talking about one particular carcinogen, PhIP, which is 2-amino-1-methyl-6-phenyl-imidazole 4,5-B pyridine. That's the most abundant mutagen by mass in cooked beef, and is responsible for 20% of the total mutagenicity. Boy, I can't say that word, sorry. Mutagenicity found in fried beef. So, McDonald's, for example, fries their beef. Daily intake of PhIP among Americans is estimated to be 280 to 460 nanograms per day per person. So there that is. Maybe we should all be vegetarians. Epigenetic events may cause some cancer. So epigenetic events are not a change in the gene sequence, but a change in the modification of the DNA such that the DNA is more or less likely to be accessed. So how it's wrapped up on the chromatin makes it more likely or less likely to be expressed. So there are examples like, for example, P16 is a possible oncogene, but it can be not an oncogene, not mutated, but heavily methylated in many cancers. So it affects its ability to be transcribed, which is essentially a similar effect as if it were mutated. Is there a link between cancer and electromotive force? So this is the idea that you shouldn't live near power lines, or you shouldn't have electric blankets because you're putting yourself under an electrical field. So I've looked at a fair number of studies about this. If you look at a cell-based assay, something that looks at cell culture, and then you look at things like how many mutations in the DNA result as a result of being exposed to electromagnetic force, you can almost always see an effect. But if you look at epidemiological studies where they look at large number of people and their proximity to a power line or a power source or something like that, they typically don't show an effect. That there's no significant difference that's associated with someone who does or does not live near a power line. So in addition to DNA mutation effects in a cell-based assay, you might see increase in calcium signaling or increase in RNA synthesis or changes in gene expression, increase in activity of a kinase. But in general, no effects on DNA mutation. I did say that's what they were looking for, but typically don't see an effect on DNA mutation. Or big chromosomal breaks, like in Burkitt's lymphoma we talked about, or in how much nitric oxide is produced, or the likelihood of tumors being produced in cell culture, or in animal models, or in changes in DNA synthesis, just the entering the S phase, in other words, cell proliferation, those sorts of things. In studies where some link was found, they try to repeat it using the same conditions, and 11 out of 13 times, and they try to repeat it, they can't repeat it. So if there is something there, it is not obvious or repeatable. Okay, another H5P question. All right. What about cancer and cell phones? So we all have cell phones. We use them a lot. There's been lots of speculation about if cancer causes increase in or if cell phone use causes an increase in cancer rate. So International Journal of Oncology did a meta-study in 2008. It's a study of other studies where they looked at similar methods and tried to use statistics to find out is there an overall effect or not, looking at many different studies. And they found that 10 years of exposure doubles the risk of malignant brain tumors on the same side of the brain, but not the opposite side of the brain. So if you hold your phone up to your right ear, if you're right-handed, and you've been doing that for 10 years, your risk of a malignant brain tumor on that side of your head versus the other side of the head is twice as high. And 2.5 times increased risk of acoustic neuromas, which are benign auditory nerve tumors. So that seems pretty strong evidence, but remember, like double the risk could mean 1 in a million to now 2 in a million. For kids, it may be a bigger problem because they have a thinner skull, and so any kind of electromagnetic radiation produced by the phone is going to penetrate through the skull easier. There's a study in 2012 in bioelectromagnetics that showed no increased risk for intracranial tumors. One in 2014, these are since 2008, which showed no increased risk, and one in 2016, also no increased risk. So most of you guys are not using your phone to talk on anyway. I think you should be more concerned if there is any effect on this on your thumbs from texting. But it doesn't look like there's a strong association based on the more recent data. Okay, other things where there is a strong association, like in breast cancer. So 2 genes, BRCA1 and BRCA2, have been identified for a long time, and they as giving people who express these oncogenes a much higher probability of developing breast cancer and or ovarian cancer sometime during their life if they're mutated. So there's certain mutations within BRCA1 or BRCA2 that can increase someone's chance pretty dramatically, maybe 4 times higher than a person without these mutations. There have been some studies where they used chemotherapy on healthy women, and even though they're healthy women, they don't have breast cancer, but they have these risk factor genes, and it reduced their their incidence of breast cancer by 45%. But these are not happy drugs. These are the general chemotherapy drugs that have big side effects, like nausea and hair loss. They also have the side effect of increasing endometrial cancer, so you reduce the incidence of breast cancer, but you increase the risk of endometrial cancer, and also blood clots. But those increased risks only are a problem if it's typically after menopause, after age 50, which for most women is after menopause. So the genes were identified before we knew what their function was. The function of the BRCA genes has only come out in the last couple of years. We know that they are DNA repair mechanisms. They repair double-stranded breaks by homologous recombination. And here is ATM that we talked about earlier in this podcast that detects double-stranded breaks, activate the BRCA genes to repair these double-stranded breaks. Not too hard to imagine that if you can't repair those breaks and instead you send cells into the S phase with double-stranded breaks, that you're going to get mutations. So there's some interesting social biology around BRCA genes and interaction with science and legality, and that is, can you own a gene? Can you patent a gene? So getting tested for whether or not a person is carrying this gene is very important for the women who are worried about this as a risk factor. They may be advised to get tested based on their family histories. For example, if there have been other women in their family who've had breast cancer. And the test has been expensive, $3,000. And so that may be prohibitive for a lot of women, and a lot of women are excluded from knowing if they have this increased risk. So there were court cases against Myriad Genetics. These are the people who patented the gene, and they were the people charging $3,000 for the test. And the test is a very simple test. I mean, if you've been in lab, you've done the equivalent in that you extract DNA from the people. You can extract it from any source. So you can do a cheek swab. You use primers on that flank this region of the BRCA gene, and you sequence that. And so it doesn't cost $3,000 to do that. The sequencing only costs about less than $5. And then if you add in all the other stuff, you could definitely do it for less than $50, even with shipping and all that sort of stuff. So in the first court case, there was a brief filed that said, to allow patent on a gene isolated from the body is akin to allowing the first surgeon who removed a kidney to patent any and all isolated kidneys. The holder of the patent on an isolated kidney could prevent other surgeons from removing isolating kidney diseases. The patent holder could also use its exclusive rights to charge a royalty of $3,000 or more each time a person donated a kidney to a relative. So obviously that's ridiculous, and they're saying that's essentially what Myriad Genetics is saying they're doing, since we all have these genes, males and females. All humans have these genes. How can anyone patent a gene? And certainly there was nothing patentable about their test, because they were just doing PCR, which they don't own the patent on. They lost in the U.S. Supreme Court, and then there was an appeal. It lost in that they couldn't patent the gene. 2012, there was an appeal, and their argument was they made a cDNA library, which is not a natural phenomenon. So when you make cDNA, that does not happen naturally inside of a cell, and so they're saying that was a unique, patentable experience as part of the process, and therefore they should be able to patent that. But in 2013, the Supreme Court ruled that a naturally occurring DNA segment is a product of nature and not patent eligible merely because it has been isolated. But the cDNA part might be patentable. They also tried this in other countries, like in Australia. In Australia, they lost their case also. So there is no patent now in the U.S. allowed on individual human genes. Doesn't mean you can't patent a test that uses a gene, but there has to be something about your procedure, something you made or something you did that is unique and patentable. You can't just say, we have the patent on this gene or sequencing that gene. I just wanted to mention this in case anybody out there is thinking about getting a BRCA test. So Myriad Genetics is still charging a bunch. It's over $1,000, I think, for their test. And 23andMe started advertising, I think last year or 2 years ago, that they're offering BRCA tests. And 23andMe, you can get supposedly your whole genome done for $100, so that's lots cheaper. And a lot of women decided to get tested for their genome at 23andMe simply to find out if they were if they had concern for the BRCA mutations. The problem is that BRCA is not, or 23andMe is not actually sequencing your DNA. They're just looking for specific single nucleotide polymorphisms, called SNPs, that you may or may not express. And the 23andMe panel that looks at thousands of these SNPs in anybody's DNA, for BRCA1 and BRCA2, they're only looking at 3 possible mutations that humans express. but there are over 1,000 mutations that are known in GenBank for BRCA mutations. And if someone got a test result back from 23andMe saying that you are clear for these BRCA mutations, that could give them a false sense of not having to be concerned about this in their future, and not be vigilant about their own health. And they weren't actually clear because they had one of the other 997 known variations. So they are 23andMe is being misleading about what they're providing in terms of this service and how much it costs. So I've encouraged people in my life who are getting this test to try to get their insurance to pay for the expensive one, because then you'll know at least what the edge of science is at the time where you have the test, and all known mutations at that time. And the whole reason you're getting the test is so you have some idea about what your risk is, and so find out what your real risk is, and not don't be deceived by this 23andMe test. Okay, this is an old figure, 2008, but even so, it makes the point that everything in this generalized diagram of an animal cell that is in red is a known oncogene. And there are lots on here, and this is nowhere near complete of all the known oncogenes we do know. And so the idea that we can find a general cure-all for cancer is, for no matter what kind of cancer it is pretty far-fetched, considering all these different pathways. But the idea that we can increase our monitoring for people's possible cancer, and to detect those cancers before they become metastatic, and while they're still treatable in a way that is not debilitating to the person, that is not far-fetched. And in fact, as you saw on the cancer.gov site, the rate of cancer deaths has gone down. The news on cancer is relatively good going forward, and certainly making lots of progress in cancer research. So even though it's a big problem and a daunting problem, science has made tremendous headway in terms of cancer practice and cancer research. Okay, that's it.